Chapter 8. Energy and Metabolism. Concept Outline. Section 8.1. The laws of thermodynamics describe how energy changes. The flow of energy in living things. Potential energy is present in the valence electrons of atoms, and so can be transferred from one molecule to another. The laws of thermodynamics. Energy is never lost, but as it is transferred, more and more of it dissipates as heat, a disordered form of energy. Free energy. In a chemical reaction, the energy released or supplied is the difference in bond energies between reactants and products, corrected for disorder. Activation energy. To start a chemical reaction, an input of energy is required to destabilize existing chemical bonds. Section 8.2. Enzymes are biological catalysts. Enzymes. Globular proteins called enzymes catalyze chemical reactions within cells. How enzymes work. Enzymes have sites on their surface shaped to fit their substrates snugly, forcing chemically reactive groups close enough to facilitate a reaction. Enzymes take many forms. Some enzymes are associated in complex groups, others are not even proteins. Factors affecting enzyme activity. Each enzyme works most efficiently at its optimal temperature and pH. Metal ions or other substances often help enzymes carry out their catalysis. Section 8.3. ATP is the energy currency of life. What is ATP? Cells store and release energy from the phosphate bonds of ATP, the energy currency of the cell. Section 8.4. Metabolism is the chemical life of a cell. Biochemical pathways, the organizational units of metabolism. Biochemical pathways are the organizational units of metabolism. The evolution of metabolism. The major metabolic processes evolved over a long period, building on what came before. Life can be viewed as a constant flow of energy, channeled by organisms to do the work of living. Each of the significant properties by which we define life, order, growth, reproduction, responsiveness, and internal regulation, requires a constant supply of energy, figure 8.1. Deprived of a source of energy, life stops. Therefore, a comprehensive study of life would be impossible without discussing bioenergetics, the analysis of how energy powers the activities of living systems. In this chapter, we will focus on energy, on what it is and how organisms capture, store, and use it. Section 8.1. The laws of thermodynamics describe how energy changes. The flow of energy in living things. Energy is defined as the capacity to do work. It can be considered to exist in two states. Kinetic energy is the energy of motion, figure 8.2. Moving objects perform work by causing other matter to move. Potential energy is stored energy. Objects that are not actively moving but have the capacity to do so possess potential energy. A boulder perched on a hilltop has potential energy. As it begins to roll downhill, some of its potential energy is converted into kinetic energy. Much of the work that living organisms carry out involves transforming potential energy to kinetic energy. Energy can take many forms: mechanical energy, heat, sound, electric current, light, or radioactive radiation. Because it can exist in so many forms, there are many ways to measure energy. The most convenient is in terms of heat, because all other forms of energy can be converted into heat. In fact, the study of energy is called thermodynamics, meaning heat changes. The unit of heat most commonly employed in biology is the kilocalorie, kcal. 1 kilocalorie is equal to 1,000 calories, cal, and 1 calorie is the heat required to raise the temperature of 1 gram of water 1 degree Celsius, degree C. It is important not to confuse calories with a term related to diets and nutrition, the calorie with a capital C, which is actually another term for kilocalorie. Another energy unit, often used in physics, is the joule. 1 joule equals 0.239 calories. Oxidation reduction. Energy flows into the biological world from the sun, which shines a constant beam of light on the Earth. It is estimated that the Sun provides the Earth with more than 13 by 1023 calories per year, or 40 million billion calories per second. Plants, algae, and certain kinds of bacteria capture a fraction of this energy through photosynthesis. In photosynthesis, energy garnered from sunlight is used to combine small molecules, water and carbon dioxide, into more complex molecules, sugars. The energy is stored as potential energy in the covalent bonds between atoms in the sugar molecules. Recall from Chapter 2 that an atom consists of a central nucleus surrounded by one or more orbiting electrons, and a covalent bond forms when two atomic nuclei share valence electrons. Breaking such a bond requires energy to pull the nuclei apart. Indeed, the strength of a covalent bond is measured by the amount of energy required to break it. For example, it takes 98.8 kilocalories to break 1 mole, 6.023 times 10 to the 23rd, of carbon-hydrogen (CH) bonds. During a chemical reaction, the energy stored in chemical bonds may transfer to new bonds. In some of these reactions, electrons actually pass from one atom or molecule to another. When an atom or molecule loses an electron, it is said to be oxidized, and the process by which this occurs is called oxidation. The name reflects the fact that in biological systems oxygen, which attracts electrons strongly, is the most common electron acceptor. Conversely, when an atom or molecule gains an electron, it is said to be reduced, and the process is called reduction. Oxidation and reduction always take place together, because every electron that is lost by an atom through oxidation is gained by some other atom through reduction. Therefore, chemical reactions of this sort are called oxidation-reduction, redox, reactions, figure 8.3. Energy is transferred from one molecule to another via redox reactions. The reduced form of a molecule thus has a higher level of energy than the oxidized form, figure 8.4. Oxidation-reduction reactions play a key role in the flow of energy through biological systems because the electrons that pass from one atom to another carry energy with them. The amount of energy an electron possesses depends on how far it is from the nucleus and how strongly the nucleus attracts it. Light, and other forms of energy, can add energy to an electron and boost it to a higher energy level. When this electron departs from one atom, oxidation, and moves to another, reduction, the electron's added energy is transferred with it, and the electron orbits the second atom's nucleus at the higher energy level. The added energy is stored as potential chemical energy that the atom can later release when the electron returns to its original energy level. Synopsis. Energy is the capacity to do work either actively, kinetic energy, or stored for later use, potential energy. Energy is transferred with electrons. Oxidation is the loss of an electron, reduction is the gain of one. The laws of thermodynamics. Running, thinking, singing, reading these words, all activities of living organisms involve changes in energy. A set of universal laws we call the laws of thermodynamics govern all energy changes in the universe, from nuclear reactions to the buzzing of a bee. The first law of thermodynamics. The first of these universal laws, the first law of thermodynamics, concerns the amount of energy in the universe. It states that energy cannot be created or destroyed, it can only change from one form to another, from potential to kinetic, for example. The total amount of energy in the universe remains constant. The lion eating a giraffe in figure 8.1 is in the process of acquiring energy. Rather than creating new energy or capturing the energy in sunlight, the lion is merely transferring some of the potential energy stored in the giraffe's tissues to its own body, just as the giraffe obtained the potential energy stored in the plants it ate while it was alive. Within any living organism, this chemical potential energy can be shifted to other molecules and stored in different chemical bonds, or it can convert into other forms, such as kinetic energy, light, or electricity. During each conversion, some of the energy dissipates into the environment as heat, a measure of the random motions of molecules, and, hence, a measure of one form of kinetic energy. Energy continuously flows through the biological world in one direction, with new energy from the sun constantly entering the system to replace the energy dissipated as heat. Heat can be harnessed to do work only when there is a heat gradient, that is, a temperature difference between two areas. This is how a steam engine functions. Cells are too small to maintain significant internal temperature differences, so heat energy is incapable of doing the work of cells. Thus, although the total amount of energy in the universe remains constant, the energy available to do work decreases, as progressively more of it dissipates as heat. The second law of thermodynamics. The second law of thermodynamics concerns this transformation of potential energy into heat, or random molecular motion. It states that the disorder, more formally called entropy, in the universe is continuously increasing. Put simply, disorder is more likely than order. For example, it is much more likely that a column of bricks will tumble over than that a pile of bricks will arrange themselves spontaneously to form a column. In general, energy transformations proceed spontaneously to convert matter from a more ordered, less stable form, to a less ordered, more stable form, figure 8.5. Entropy. Entropy is a measure of the disorder of a system, so the second law of thermodynamics can also be stated simply as entropy increases. When the universe formed, it held all the potential energy it will ever have. It has become progressively more disordered ever since, with every energy exchange increasing the amount of entropy. Synopsis. The first law of thermodynamics states that energy cannot be created or destroyed, it can only undergo conversion from one form to another. The second law of thermodynamics states that disorder, entropy, in the universe is increasing. As energy is used, more and more of it is converted to heat, the energy of random molecular motion. Free energy. It takes energy to break the chemical bonds that hold the atoms in a molecule together. Heat energy, because it increases atomic motion, makes it easier for the atoms to pull apart. Both chemical bonding and heat have a significant influence on a molecule, the former reducing disorder and the latter increasing it. The net effect, the amount of energy actually available to break and subsequently form other chemical bonds, is called the free energy of that molecule. In a more general sense, free energy is defined as the energy available to do work in any system. In a molecule within a cell, where pressure and volume usually do not change, the free energy is denoted by the symbol G, for Gibbs free energy, which limits the system being considered to the cell. G is equal to the energy contained in a molecule's chemical bonds, called enthalpy and designated H, minus the energy unavailable because of disorder, called entropy and given the symbol S, times the absolute temperature, T, in degrees Kelvin, K equals degree C plus 273. G equals H minus Ts. Chemical reactions break some bonds in the reactants and form new bonds in the products. Consequently, reactions can produce changes in free energy. When a chemical reaction occurs under conditions of constant temperature, pressure, and volume, as do most biological reactions, the change in free energy, delta G, is simply delta G equals delta H minus T delta S. The change in free energy, or delta G, is a fundamental property of chemical reactions. In some reactions, the delta G is positive. This means that the products of the reaction contain more free energy than the reactants, the bond energy, H, is higher or the disorder, S, in the system is lower. Such reactions do not proceed spontaneously because they require an input of energy. Any reaction that requires an input of energy is said to be endergonic, inward energy. In other reactions, the delta G is negative. The products of the reaction contain less free energy than the reactants, either the bond energy is lower or the disorder is higher, or both. Such reactions tend to proceed spontaneously. Any chemical reaction will tend to proceed spontaneously if the difference in disorder, T delta S, is greater than the difference in bond energies between reactants and products, delta H. Note that spontaneous does not mean the same thing as instantaneous. A spontaneous reaction may proceed very slowly. These reactions release the excess free energy as heat and are thus said to be exergonic, outward energy. Figure 8.6 sums up these reactions. Synopsis. Free energy is the energy available to do work. Within cells, the change in free energy, delta G, is the difference in bond energies between reactants and products, delta H, minus any change in the degree of disorder of the system, T delta S. Any reaction whose products contain less free energy than the reactants, delta G is negative, will tend to proceed spontaneously. Activation energy. If all chemical reactions that release free energy tend to occur spontaneously, why haven't all such reactions already occurred? One reason they haven't is that most reactions require an input of energy to get started. Before it is possible to form new chemical bonds, even bonds that contain less energy, it is first necessary to break the existing bonds, and that takes energy. The extra energy required to destabilize existing chemical bonds and initiate a chemical reaction is called activation energy, figure 8.7a. The rate of an exergonic reaction depends on the activation energy required for the reaction to begin. Reactions with larger activation energies tend to proceed more slowly because fewer molecules succeed in overcoming the initial energy hurdle. Activation energies are not constant, however. Stressing particular chemical bonds can make them easier to break. The process of influencing chemical bonds in a way that lowers the activation energy needed to initiate a reaction is called catalysis, and substances that accomplish this are known as catalysts, figure 8.7b. Catalysts cannot violate the basic laws of thermodynamics. They cannot, for example, make an endergonic reaction proceed spontaneously. By reducing the activation energy, a catalyst accelerates both the forward and the reverse reactions by exactly the same amount. Hence, it does not alter the proportion of reactant ultimately converted into product. To grasp this, imagine a bowling ball resting in a shallow depression on the side of a hill. Only a narrow rim of dirt below the ball prevents it from rolling down the hill. Now imagine digging away that rim of dirt. If you remove enough dirt from below the ball, it will start to roll down the hill, but removing dirt from below the ball will never cause the ball to roll up the hill. Removing the lip of dirt simply allows the ball to move freely. Gravity determines the direction it then travels. Lowering the resistance to the ball's movement will promote the movement dictated by its position on the hill. Similarly, the direction in which a chemical reaction proceeds is determined solely by the difference in free energy. Like digging away the soil below the bowling ball on the hill, catalysts reduce the energy barrier preventing the reaction from proceeding. Catalysts don't favor endergonic reactions any more than digging makes the hypothetical bowling ball roll uphill. Only exergonic reactions can proceed spontaneously, and catalysts cannot change that. What catalysts can do is make a reaction proceed much faster. Synopsis. The rate of a reaction depends on the activation energy necessary to initiate it. Catalysts reduce the activation energy and so increase the rates of reactions, although they do not change the final proportions of reactants and products. Section 8.2. Enzymes are biological catalysts. Enzymes. The chemical reactions within living organisms are regulated by controlling the points at which catalysis takes place. Life itself is, therefore, regulated by catalysts. The agents that carry out most of the catalysis in living organisms are proteins called enzymes. There is increasing evidence that some types of biological catalysis are carried out by RNA molecules. The unique three-dimensional shape of an enzyme enables it to stabilize a temporary association between substrates, the molecules that will undergo the reaction. By bringing two substrates together in the correct orientation, or by stressing particular chemical bonds of a substrate, an enzyme lowers the activation energy required for new bonds to form. The reaction thus proceeds much more quickly than it would without the enzyme. Because the enzyme itself is not changed or consumed in the reaction, only a small amount of an enzyme is needed, and it can be used over and over. As an example of how an enzyme works, let's consider the reaction of carbon dioxide and water to form carbonic acid. This important enzyme-catalyzed reaction occurs in vertebrate red blood cells. CO2 plus H2O produces H2CO3. Or carbon dioxide and water produce carbonic acid. This reaction may proceed in either direction, but because it has a large activation energy, the reaction is very slow in the absence of an enzyme. Perhaps 200 molecules of carbonic acid form in an hour in a cell. Reactions that proceed this slowly are of little use to a cell. Cells overcome this problem by employing an enzyme within their cytoplasm called carbonic anhydrase. Enzyme names usually end in ASE. Under the same conditions, but in the presence of carbonic anhydrase, an estimated 600,000 molecules of carbonic acid form every second. Thus, the enzyme increases the reaction rate more than 10 million times. Thousands of different kinds of enzymes are known, each catalyzing one or a few specific chemical reactions. By facilitating particular chemical reactions, the enzymes in a cell determine the course of metabolism, the collection of all chemical reactions, in that cell. Different types of cells contain different sets of enzymes, and this difference contributes to structural and functional variations among cell types. The chemical reactions taking place within a red blood cell differ from those that occur within a nerve cell, in part because the cytoplasm and membranes of red blood cells and nerve cells contain different arrays of enzymes. Synopsis. Cells use proteins called enzymes as catalysts to lower activation energies. How enzymes work. Most enzymes are globular proteins with one or more pockets or clefts on their surface called active sites, figure 8.8. Substrates bind to the enzyme at these active sites, forming an enzyme-substrate complex. For catalysis to occur within the complex, a substrate molecule must fit precisely into an active site. When that happens, amino acid side groups of the enzyme end up in close proximity to certain bonds of the substrate. These side groups interact chemically with the substrate, usually stressing or distorting a particular bond and consequently lowering the activation energy needed to break the bond. The substrate, now a product, then dissociates from the enzyme. Proteins are not rigid. The binding of a substrate induces the enzyme to adjust its shape slightly, leading to a better induced fit between enzyme and substrate, figure 8.9. This interaction may also facilitate the binding of other substrates. In such cases, the substrate itself activates the enzyme to receive other substrates. Synopsis. Enzymes typically catalyze only one or a few similar chemical reactions because they are specific in their choice of substrates. This specificity is due to the active site of the enzyme, which is shaped so that only a certain substrate molecule will fit into it. Enzymes take many forms. While many enzymes are suspended in the cytoplasm of cells, free to move about and not attached to any structure, other enzymes function as integral parts of cell structures and organelles. Multi-enzyme complexes. Often in cells the several enzymes catalyzing the different steps of a sequence of reactions are loosely associated with one another in non-covalently bonded assemblies called multi-enzyme complexes. The bacterial pyruvate dehydrogenase multi-enzyme complex seen in figure 8.10 contains enzymes that carry out three sequential reactions in oxidative metabolism. Each complex has multiple copies of each of the three enzymes, 60 protein subunits in all. The many subunits work in concert, like a tiny factory. Multi-enzyme complexes offer significant advantages in catalytic efficiency. 1. The rate of any enzyme reaction is limited by the frequency with which the enzyme collides with its substrate. If a series of sequential reactions occurs within a multi-enzyme complex, the product of one reaction can be delivered to the next enzyme without releasing it to diffuse away. 2. Because the reacting substrate never leaves the complex during its passage through the series of reactions, the possibility of unwanted side reactions is eliminated. 3. All of the reactions that take place within the multi-enzyme complex can be controlled as a unit. In addition to pyruvate dehydrogenase, which controls entry to the Krebs cycle, several other key processes in the cell are catalyzed by multi-enzyme complexes. One well-studied system is the fatty acid synthetase complex that catalyzes the synthesis of fatty acids from two carbon precursors. There are 7 different enzymes in this multi-enzyme complex, and the reaction intermediates remain associated with the complex for the entire series of reactions. Not all biological catalysts are proteins. Until a few years ago, most biology textbooks contain statements such as, enzymes are the catalysts of biological systems. We can no longer make that statement without qualification. As discussed in Chapter 4, Tom Cech and his colleagues at the University of Colorado reported in 1981 that certain reactions involving RNA molecules appear to be catalyzed in cells by RNA itself, rather than by enzymes. This initial observation has been corroborated by additional examples of RNA catalysis in the last few years. Like enzymes, these RNA catalysts, which are loosely called ribozymes, greatly accelerate the rate of particular biochemical reactions and show extraordinary specificity with respect to the substrates on which they act. There appear to be at least 2 sorts of ribozymes. Those that carry out intramolecular catalysis have folded structures and catalyze reactions on themselves. Those that carry out intermolecular catalysis act on other molecules without themselves being changed in the process. Many important cellular reactions involve small RNA molecules, including reactions that chip out unnecessary sections from RNA copies of genes, that prepare ribosomes for protein synthesis, and that facilitate the replication of DNA within mitochondria. In all of these cases, the possibility of RNA catalysis is being actively investigated. It seems likely, particularly in the complex process of photosynthesis, that both enzymes and RNA play important catalytic roles. The ability of RNA, an informational molecule, to act as a catalyst has stirred great excitement among biologists, as it appears to provide a potential answer to the chicken and egg riddle posed by the spontaneous origin of life hypothesis discussed in Chapter 3. Which came first, the protein or the nucleic acid? It now seems at least possible that RNA may have evolved first and catalyzed the formation of the first proteins. Synopsis. Not all biological catalysts float free in the cytoplasm. Some are part of other structures, and some are not even proteins. Factors affecting enzyme activity. The rate of an enzyme-catalyzed reaction is affected by the concentration of substrate, and of the enzyme that works on it. In addition, any chemical or physical factor that alters the enzyme's three-dimensional shape, such as temperature, pH, salt concentration, and the binding of specific regulatory molecules, can affect the enzyme's ability to catalyze the reaction. Temperature. Increasing the temperature of an uncatalyzed reaction will increase its rate because the additional heat represents an increase in random molecular movement. The rate of an enzyme-catalyzed reaction also increases with temperature, but only up to a point called the temperature optimum, figure 8.11a. Below this temperature, the hydrogen bonds and hydrophobic interactions that determine the enzyme's shape are not flexible enough to permit the induced fit that is optimum for catalysis. Above the temperature optimum, these forces are too weak to maintain the enzyme's shape against the increased random movement of the atoms in the enzyme. At these higher temperatures, the enzyme denatures, as we described in Chapter 3. Most human enzymes have temperature optima between 35 degrees Celsius and 40 degrees Celsius, a range that includes normal body temperature. Bacteria that live in hot springs have more stable enzymes, that is, enzymes held together more strongly, so the temperature optima for those enzymes can be 70 degrees Celsius or higher. pH. Ionic interactions between oppositely charged amino acid residues, such as glutamic acid, and lysine, also hold enzymes together. These interactions are sensitive to the hydrogen ion concentration of the fluid the enzyme is dissolved in, because changing that concentration shifts the balance between positively and negatively charged amino acid residues. For this reason, most enzymes have a pH optimum that usually ranges from pH 6 to 8. Those enzymes able to function in very acidic environments are proteins that maintain their three-dimensional shape even in the presence of high levels of hydrogen ion. The enzyme pepsin, for example, digests proteins in the stomach at pH 2, a very acidic level, figure 8.11b. Inhibitors and activators. Enzyme activity is sensitive to the presence of specific substances that bind to the enzyme and cause changes in its shape. Through these substances, a cell is able to regulate which enzymes are active and which are inactive at a particular time. This allows the cell to increase its efficiency and to control changes in its characteristics during development. A substance that binds to an enzyme and decreases its activity is called an inhibitor. Very often, the end product of a biochemical pathway acts as an inhibitor of an early reaction in the pathway, a process called feedback inhibition, to be discussed later. Enzyme inhibition occurs in two ways. Competitive inhibitors compete with the substrate for the same binding site, displacing a percentage of substrate molecules from the enzymes. Non-competitive inhibitors bind to the enzyme in a location other than the active site, changing the shape of the enzyme and making it unable to bind to the substrate, figure 8.12. Most non-competitive inhibitors bind to a specific portion of the enzyme called an allosteric site, Greek allos, other, plus steros, form. These sites serve as chemical on-off switches. The binding of a substance to the site can switch the enzyme between its active and inactive configurations. A substance that binds to an allosteric site and reduces enzyme activity is called an allosteric inhibitor, figure 8.12b. Alternatively, activators bind to allosteric sites and keep the enzymes in their active configurations, thereby increasing enzyme activity. Enzyme cofactors. Enzyme function is often assisted by additional chemical components known as cofactors. For example, the active sites of many enzymes contain metal ions that help draw electrons away from substrate molecules. The enzyme carboxypeptidase digests proteins by employing a zinc ion, Zn++, in its active site to remove electrons from the bonds joining amino acids. Other elements, such as molybdenum and manganese, are also used as cofactors. Like zinc, these substances are required in the diet in small amounts. When the cofactor is a non-protein organic molecule, it is called a coenzyme. Many vitamins are parts of coenzymes. In numerous oxidation-reduction reactions that are catalyzed by enzymes, the electrons pass in pairs from the active site of the enzyme to a coenzyme that serves as the electron acceptor. The coenzyme then transfers the electrons to a different enzyme, which releases them, and the energy they bear, to the substrates in another reaction. Often, the electrons pair with protons, H+, as hydrogen atoms. In this way, coenzymes shuttle energy in the form of hydrogen atoms from one enzyme to another in a cell. One of the most important coenzymes is the hydrogen acceptor nicotinamide adenine dinucleotide, NAD+, figure 8.13. The NAD+ molecule is composed of two nucleotides bound together. As you may recall from chapter 3, a nucleotide is a 5-carbon sugar with one or more phosphate groups attached to one end and an organic base attached to the other end. The two nucleotides that make up NAD+, nicotinamide monophosphate, NMP, and adenine monophosphate, AMP, are joined head-to-head by their phosphate groups. The two nucleotides serve different functions in the NAD+ molecule. AMP acts as the core, providing a shape recognized by many enzymes. NMP is the active part of the molecule, contributing a site that is readily reduced, that is, easily accepts electrons. When NAD+ acquires an electron and a hydrogen atom, actually, 2 electrons and a proton, from the active site of an enzyme, it is reduced to NADH. The NADH molecule now carries the 2 energetic electrons and the proton. The oxidation of energy-containing molecules, which provides energy to cells, involves stripping electrons from those molecules and donating them to NAD+. As we'll see, much of the energy of NADH is transferred to another molecule. Synopsis. Enzymes have an optimum temperature and pH, at which the enzyme functions most effectively. Inhibitors decrease enzyme activity, while activators increase it. The activity of enzymes is often facilitated by cofactors, which can be metal ions or other substances. Cofactors that are non-protein organic molecules are called coenzymes. Catalysis, a closer look at carbonic anhydrase. One of the most rapidly acting enzymes in the human body is carbonic anhydrase, which plays a key role in blood by converting dissolved CO2 into carbonic acid, which dissociates into bicarbonate and hydrogen ions. CO2 plus H2O produces H2CO3 produces HCO3- plus H+. Fully 70% of the CO2 transported by the blood is transported as bicarbonate ion. This reaction is exergonic, but its energy of activation is significant, so that little conversion to bicarbonate occurs spontaneously. In the presence of the enzyme carbonic anhydrase, however, the rate of the reaction accelerates by a factor of more than 10 million. How does carbonic anhydrase catalyze this reaction so effectively? The active site of the enzyme is a deep cleft traversing the enzyme, as if it had been cut with the blade of an axe. Deep within the cleft, some 1.5 nanometers from the surface, are located 3 histidines, their imidazole, nitrogen ring, groups all pointed at the same place in the center of the cleft. Together they hold a zinc ion firmly in position. This zinc ion will be the cutting blade of the catalytic process. Here is how the zinc catalyzes the reaction. Immediately adjacent to the position of the zinc atom in the cleft are a group of amino acids that recognize and bind carbon dioxide. The zinc atom interacts with this carbon dioxide molecule, orienting it in the plane of the cleft. Meanwhile, water bound to the zinc is rapidly converted to hydroxide ion. This hydroxide ion is now precisely positioned to attack the carbon dioxide. When it does so, HCO3- is formed, and the enzyme is unchanged, figure 8a. Carbonic anhydrase is an effective catalyst because it brings its 2 substrates into close proximity and optimizes their orientation for reaction. Other enzymes use other mechanisms. Many, for example, use charged amino acids to polarize substrates or electronegative amino acids to stress particular bonds. Whatever the details of the reaction, however, the precise positioning of substrates achieved by the particular shape of the enzyme. Section 8.3. ATP is the energy currency of life. What is ATP? The chief energy currency all cells use is a molecule called adenosine triphosphate, ATP. Cells use their supply of ATP to power almost every energy-requiring process they carry out, from making sugars, to supplying activation energy for chemical reactions, to actively transporting substances across membranes, to moving through their environment and growing. Structure of the ATP molecule. Each ATP molecule is a nucleotide composed of 3 smaller components, figure 8.14. The first component is a 5-carbon sugar, ribose, which serves as the backbone to which the other 2 subunits are attached. The second component is adenine, an organic molecule composed of 2 carbon-nitrogen rings. Each of the nitrogen atoms in the ring has an unshared pair of electrons and weakly attracts hydrogen ions. Adenine, therefore, acts chemically as a base and is usually referred to as a nitrogenous base. It is one of the 4 nitrogenous bases found in DNA and RNA. The third component of ATP is a triphosphate group, a chain of 3 phosphates. How ATP stores energy. The key to how ATP stores energy lies in its triphosphate group. Phosphate groups are highly negatively charged, so they repel one another strongly. Because of the electrostatic repulsion between the charged phosphate groups, the 2 covalent bonds joining the phosphates are unstable. The ATP molecule is often referred to as a coiled spring, the phosphates straining away from one another. The unstable bonds holding the phosphates together in the ATP molecule have a low activation energy and are easily broken. When they break, they can transfer a considerable amount of energy. In most reactions involving ATP, only the outermost high-energy phosphate bond is hydrolyzed, cleaving off the phosphate group on the end. When this happens, ATP becomes adenosine diphosphate, ADP, and energy equal to 7.3 kilocalories per mole is released under standard conditions. The liberated phosphate group usually attaches temporarily to some intermediate molecule. When that molecule is dephosphorylated, the phosphate group is released as inorganic phosphate, PI. How ATP powers energy-requiring reactions. Cells use ATP to drive endergonic reactions. Such reactions do not proceed spontaneously, because their products possess more free energy than their reactants. However, if the cleavage of ATP's terminal high-energy bond releases more energy than the other reaction consumes, the overall energy change of the two coupled reactions will be exergonic, energy-releasing, and they will both proceed. Because almost all endergonic reactions require less energy than is released by the cleavage of ATP, ATP can provide most of the energy a cell needs. The same feature that makes ATP an effective energy donor, the instability of its phosphate bonds, precludes it from being a good long-term energy storage molecule. Fats and carbohydrates serve that function better. Most cells do not maintain large stockpiles of ATP. Instead, they typically have only a few seconds supply of ATP at any given time, and they continually produce more from ADP and PI. Synopsis. The instability of its phosphate bonds makes ATP an excellent energy donor. Section 8.4. Metabolism is the chemical life of a cell. Biochemical pathways, the organizational units of metabolism. This living chemistry, the total of all chemical reactions carried out by an organism, is called metabolism, Greek metabol, change. Those reactions that expend energy to make or transform chemical bonds are called anabolic reactions, or anabolism. Reactions that harvest energy when chemical bonds are broken are called catabolic reactions, or catabolism. Organisms contain thousands of different kinds of enzymes that catalyze a bewildering variety of reactions. Many of these reactions in a cell occur in sequences called biochemical pathways. In such pathways, the product of one reaction becomes the substrate for the next, figure 8.15. Biochemical pathways are the organizational units of metabolism, the elements an organism controls to achieve coherent metabolic activity. Most sequential enzyme steps in biochemical pathways take place in specific compartments of the cell. The steps of the citric acid cycle, chapter 9, for example, occur inside mitochondria. By determining where many of the enzymes that catalyze these steps are located, we can map out a model of metabolic processes in the cell. How biochemical pathways evolved. In the earliest cells, the first biochemical processes probably involved energy-rich molecules scavenged from the environment. Most of the molecules necessary for these processes are thought to have existed in the organic soup of the early oceans. The first catalyzed reactions are thought to have been simple, one-step reactions that brought these molecules together in various combinations. Eventually, the energy-rich molecules became depleted in the external environment, and only organisms that had evolved some means of making those molecules from other substances in the environment could survive. Thus, a hypothetical reaction. F plus G produces H. Where two energy-rich molecules, F and G, react to produce compound H and release energy, became more complex when the supply of F in the environment ran out. A new reaction was added in which the depleted molecule, F, is made from another molecule, E, which was also present in the environment. E produces F plus G produces H. When the supply of E in turn became depleted, organisms that were able to make it from some other available precursor, D, survived. When D became depleted, those organisms in turn were replaced by ones able to synthesize D from another molecule, C. Equation: C produces D produces E produces F plus G produces H. This hypothetical biochemical pathway would have evolved slowly through time, with the final reactions in the pathway evolving first and earlier reactions evolving later. Looking at the pathway now, we would say that the organism, starting with compound C, is able to synthesize H by means of a series of steps. This is how the biochemical pathways within organisms are thought to have evolved, not all at once, but one step at a time, backward. How biochemical pathways are regulated. For a biochemical pathway to operate efficiently, its activity must be coordinated and regulated by the cell. Not only is it unnecessary to synthesize a compound when plenty is already present, doing so would waste energy and raw materials that could be put to use elsewhere. It is, therefore, advantageous for a cell to temporarily shut down biochemical pathways when their products are not needed. The regulation of simple biochemical pathways often depends on an elegant feedback mechanism. The end product of the pathway binds to an allosteric site on the enzyme that catalyzes the first reaction in the pathway. In the hypothetical pathway we just described, the enzyme catalyzing the reaction CD would possess an allosteric site for H, the end product of the pathway. As the pathway churned out its product and the amount of H in the cell increased, it would become increasingly likely that one of the H molecules would encounter the allosteric site on the CD enzyme. If the product H functioned as an allosteric inhibitor of the enzyme, its binding to the enzyme would essentially shut down the reaction CD. Shutting down this reaction, the first reaction in the pathway, effectively shuts down the whole pathway. Hence, as the cell produces increasing quantities of the product H, it automatically inhibits its ability to produce more. This mode of regulation is called feedback inhibition, figure 8.16. Synopsis. A biochemical pathway is an organized series of reactions, often regulated as a unit. The evolution of metabolism. Metabolism has changed a great deal as life on Earth has evolved. This has been particularly true of the reactions organisms use to capture energy from the sun to build organic molecules, anabolism, and then break down organic molecules to obtain energy, catabolism. These processes, the subject of the next two chapters, evolved in concert with each other. Degradation. The most primitive forms of life are thought to have obtained chemical energy by degrading, or breaking down, organic molecules that were abiotically produced. The first major event in the evolution of metabolism was the origin of the ability to harness chemical bond energy. At an early stage, organisms began to store this energy in the bonds of ATP, an energy carrier used by all organisms today. Glycolysis. The second major event in the evolution of metabolism was glycolysis, the initial breakdown of glucose. As proteins evolved diverse catalytic functions, it became possible to capture a larger fraction of the chemical bond energy in organic molecules by breaking chemical bonds in a series of steps. For example, the progressive breakdown of the 6-carbon sugar glucose into 3-carbon molecules is performed in a series of 10 steps that results in the net production of 2 ATP molecules. The energy for the synthesis of ATP is obtained by breaking chemical bonds and forming new ones with less bond energy, the energy difference being channeled into ATP production. This biochemical pathway is called glycolysis. Glycolysis undoubtedly evolved early in the history of life on Earth, since this biochemical pathway has been retained by all living organisms. It is a chemical process that does not appear to have changed for well over 3 billion years. Anaerobic photosynthesis. The third major event in the evolution of metabolism was anaerobic photosynthesis. Early in the history of life, some organisms evolved a different way of generating ATP, called photosynthesis. Instead of obtaining energy for ATP synthesis by reshuffling chemical bonds, as in glycolysis, these organisms developed the ability to use light to pump protons out of their cells, and to use the resulting proton gradient to power the production of ATP, a process called chemiosmosis. Photosynthesis evolved in the absence of oxygen and works well without it. Dissolved H2S, present in the oceans beneath an atmosphere free of oxygen gas, served as a ready source of hydrogen atoms for building organic molecules. Free sulfur was produced as a byproduct of this reaction. Nitrogen fixation. Nitrogen fixation was the fourth major step in the evolution of metabolism. Proteins and nucleic acids cannot be synthesized from the products of photosynthesis because both of these biologically critical molecules contain nitrogen. Obtaining nitrogen atoms from N2 gas, a process called nitrogen fixation, requires the breaking of an NN triple bond. This important reaction evolved in the hydrogen-rich atmosphere of the early Earth, an atmosphere in which no oxygen was present. Oxygen acts as a poison to nitrogen fixation, which today occurs only in oxygen-free environments, or in oxygen-free compartments within certain bacteria. Oxygen-forming photosynthesis. The substitution of H2O for H2S in photosynthesis was the fifth major event in the history of metabolism. Oxygen-forming photosynthesis employs H2O rather than H2S as a source of hydrogen atoms and their associated electrons. Because it garners its hydrogen atoms from reduced oxygen rather than from reduced sulfur, it generates oxygen gas rather than free sulfur. More than 2 billion years ago, small cells capable of carrying out this oxygen-forming photosynthesis, such as cyanobacteria, became the dominant forms of life on Earth. Oxygen gas began to accumulate in the atmosphere. This was the beginning of a great transition that changed conditions on Earth permanently. Our atmosphere is now 20.9% oxygen, every molecule of which is derived from an oxygen-forming photosynthetic reaction. Aerobic respiration. Aerobic respiration is the sixth and final event in the history of metabolism. This cellular process harvests energy by stripping energetic electrons from organic molecules. Aerobic respiration employs the same kind of proton pumps as photosynthesis, and is thought to have evolved as a modification of the basic photosynthetic machinery. However, the hydrogens and their associated electrons are not obtained from H2S or H2O, as in photosynthesis, but rather from the breakdown of organic molecules. Biologists think that the ability to carry out photosynthesis without H2S first evolved among purple non-sulfur bacteria, which obtain their hydrogens from organic compounds instead. It was perhaps inevitable that among the descendants of these respiring photosynthetic bacteria, some would eventually do without photosynthesis entirely, subsisting only on the energy and hydrogens derived from the breakdown of organic molecules. The mitochondria within all eukaryotic cells are thought to be their descendants. Synopsis. 6 major innovations highlight the evolution of metabolism as we know it today. A vocabulary of metabolism. Activation energy. The energy required to destabilize chemical bonds and to initiate a chemical reaction. Catalysis. Acceleration of the rate of a chemical reaction by lowering the activation energy. Coenzyme. A non-protein organic molecule that plays an accessory role in enzyme-catalyzed reactions, often by acting as a donor or acceptor of electrons. NAD+ is a coenzyme. Endergonic reaction. A chemical reaction to which energy from an outside source must be added before the reaction proceeds, the opposite of an exergonic reaction. Entropy. A measure of the randomness or disorder of a system. In cells, it is a measure of how much energy has become so dispersed, usually as evenly distributed heat, that it is no longer available to do work. Exergonic reaction. An energy-yielding chemical reaction. Exergonic reactions tend to proceed spontaneously, although activation energy is required to initiate them. Free energy. Energy available to do work. Kilocalorie. 1000 calories. A calorie is the heat required to raise the temperature of 1 gram of water by 1 degree Celsius. Metabolism. The sum of all chemical processes occurring within a living cell or organism. Oxidation. The loss of an electron by an atom or molecule. It occurs simultaneously with reduction of some other atom or molecule because an electron that is lost by one is gained by another. Reduction. The gain of an electron by an atom or molecule. Oxidation-reduction reactions are an important means of energy transfer within living systems. Substrate. A molecule on which an enzyme acts, the initial reactant in an enzyme-catalyzed reaction.